Okay, thank you very much for joining us in this webinar, stakeholder webinar on the recently released reflection paper on the qualification of non-mutagenic impurities. This reflection paper is currently open for public consultation, and we would really recommend or ask you to provide any comments that you may have on this reflection paper using the link on the website, so using the form for public consultation. Some housekeeping. We have for this webinar, we have disabled the chat, and we have also disabled the microphones and cameras for the people that are not presenting. Any questions that you may have during the webinar, please write them down and send them through the public consultation. Some questions were submitted in advance, and they will be discussed at the last part of this webinar in the question and answer session. Between the discussion of the reflection paper and the question and answer session, we have a short coffee break of 10 to 15 minutes, depending on the time that we need for the explanation of the webinar. The webinar will be recorded and will be put together with the slides on the EMA website after this webinar has been finished. And then I would like to go to the next slide where we have the agenda for today. We will open the event. I will shortly introduce the presenters. They will go into detail on the reflection paper and the considerations that they have taken to update this reflection paper. Then we have a short coffee break. We will have some time for question answers, and we will have a wrap up and closure. So I'm happy to introduce to you the moderator of this meeting and some of the speakers, which is Leon van Aert is today our moderator. He's a former member of the MEB and the main author of this reflection paper. Then we have Marianne Schmidt from the Danish authority. She's the chair of the drafting group at the moment. We have Roland Frutschel from BFARM, and we have Dominique Massin from the French authorities. And I would like to give over the floor to Leon.
Okay, thank you, Siska. So first, the objectives of this webinar is to explain the content and the main changes implemented in the updated draft reflection paper, and also to illustrate the ongoing public consultation and providing guidance on how stakeholders can contribute. And finally, to address questions and requests for clarification received from stakeholders. Next slide, please. So these are the number of registrants for this webinar. As you can see, the main portion from industry, both generic and pharma biotech, but also a lot of registrants from the national authorities and non-EU national authorities and other EU institutions. So there is a broad interest in this webinar. Next slide, please. So this is about the update of the reflection paper, the purpose and the overview of the content and proposed changes. Next slide. So in 2017, a first draft on the reflection paper was issued, at that time still titled Qualification of Non-Genotoxic Impurities. We received stakeholders' comments on that draft reflection paper. And since issuing that draft, stakeholders have also published scientific papers, and that all took place in the meantime. After the pandemic, a new drafting group was installed and took up to redraft the reflection paper thoroughly. So it was not only making a final version, but we thought the changes are too big to not to reconsult the stakeholders, and therefore a new draft of the reflection paper has been issued now and is still open for consultation until I believe the end of April, if I'm correct. Next slide, please. So the main sections of the reflection paper are introduction, scope, and key considerations. And the main content really is about key considerations. We will address the general outline for risk assessment of non-mutagenic impurities, level of concern considerations, acceptable level calculations, new approach methodologies, API-like versus non-API-like structures, metabolites, in vivo quantification studies, and the issue of products under clinical development. And as you may have seen, all these issues are also included in the reflection paper. So what was the historical background? So the first draft was released in 2018. Sorry, I just said 17, it was 18. And we received a lot of comments from the stakeholders, and the main comments concerned that they ask for more clarity on exceeding Q3AB limits during early development, the use of 1 milligram as a limit for all drug products under 2 gram, and to provide guidance on the acceptance of alternative methods, to replace in vivo qualification studies when these are required, to maximize the results from in vivo studies used to show safety of the drug substance for qualifying impurities, and consideration of the risk benefit of the pharmaceutical for the qualification process. We believe that the current draft reflection paper on the qualification of non-mutagenic impurities addresses all of these comments that were received on basis of the draft of the previous version. Next slide, please. And next click, please. Thank you. So there were several stakeholder publications. This is the one from Harvey et al. That was already available at the time the previous version was issued. This paper focuses on early clinical trials, indicating that this is not regulated by Q3A and B. Nevertheless, there are a lot of requests from regulatory authorities on impurity levels in products under development. It was the Q3A indicates that for drugs when they are below 666 milligram, actually 1 milligram per day is the qualification threshold, and that was used as a starting point for the proposals in that paper. And they also applied a modified Haber's law to transform to short-term use, and therefore they concluded that 5 milligram per day as a qualification threshold for early clinical trials could be used. Next slide, please. Yeah, thank you. Oh, one back, please. Yeah, thank you. Then we have a paper from Graham et al. It's based on a pharmaceutical industry-wide survey by the IQ Consortium on the current impurity qualification practices. Describes that generally the drug substance NOAL is used to set specification limits for impurities, and therefore to qualify these impurities. It argues that the authors do not favor the use of body surface correction or human equivalent dose conversion. And they reviewed 181 intermediates and starting materials they call INTs. Of note, in table 2, and I think this is perhaps important for the discussions, when a maximum daily dose is taken of 2 gram, and this data is taken from table 2 in that paper, then 80% of the cases, the safety margin would be less than 100, and 53% of the cases, the safety margin would be less than 500. Next slide, please. Yeah, one click more. We have the paper from Lorty That's a survey from FPA on 13 case studies of non-mutagenic impurity qualifications among 6 member companies. And this paper clearly also demonstrates the variations in companies' strategies and also differences between regulatory acceptance of different proposals for qualification of impurities. Next slide, please. And then we have the paper from Mitra, which builds on the previous paper from Graham, the IQ Consortium survey, and in this paper, recommendations for impurity qualification study design are provided. Next slide, please. And the paper from Kenyon goes into durational aspects of the toxicity of compounds. They actually reviewed almost 5,000 chemicals and studied the distribution of the NOAELs. And they argue that the data support that 1 milligram per day is a safe threshold for non-mutagenic impurity in pharmaceuticals. And they also suggest that non-mutagenic impurity levels less than 20 milligram per day and less than 5 milligram per day are considered safe in clinical trials with less than 1 month and 1 month up to 6 months duration, respectively, would be considered safe. Next slide, please. Then we have the paper from Hasselgren, another extensive analysis of the NOAELs and other points of departures for 2,213 chemicals in this case. And it focuses more on trying to identify which chemical classes actually are the classes which contain the most potent structural compounds. And they indicate that 0.6% will have a point of departure less than 0.02 milligram per kilogram per day. They label that as very potent, and 2.4% have a POD less than 0.2 milligram per kilogram per day. Again, here in this paper, it is argued that, well, that indicates that 1 milligram per day could be acceptable for non-mutagenic impurities in general. However, there are structural classes which contain very potent compounds, including the organic thiophosphates, polychlorinated benzenes, polychlorinated polycyclic aliphatics, and also some likely potent classes like cyclic aliphatic organothiol, steroids, derivatives, and inorganic chlorine oxides. However, also a considerable number of miscellaneous potent compounds, which do not really represent a full class, but are more single compounds, which happen to have a low POD also. Next slide, please. And then the paper from survey, it's again an FPA survey, but this time a more extensive survey. It reviews 467 non-mutagenic impurities, and it demonstrates that the majority of impurities studied are studied in spiked drug substance batches, and only 1.3% is studied as a neat impurity. These studies in the spiked drug substance batches or the non-spiked drug substance batches do not provide a unique safety signal for these impurities. And again, here it is proposed that 5 milligram per day and 1 milligram per day should be acceptable as qualification thresholds for clinical trials less than 6 months and for lifelong exposure, respectively. Next slide, please. Publications in general indicate that strategies for qualification and risk assessments vary, and that 1 milligram per day would be considered as a safe threshold, and 5 milligram per day for early clinical trials up to 6 months. Testing in 28-day rat study is the most common study design using drug substance batches, which can be spiked or not spiked. And these studies do not detect concerning signals. We acknowledge that these stakeholder papers provide relevant information and are valuable when considering strategies for qualification of non-mutagenic impurities. However, the NOAELs in animal studies is considered safe for human exposure following the reasoning in these papers without consistent application of modifying factors to allow for inter-individual variation, for species differences, for study duration, seriousness of toxicity, the correction when the PUD is not a NOAEL, and route-to-route extrapolations. That does not mean that in the reasoning in none of the papers any of these aspects has been considered. However, in their argument that 1 milligram per day for humans could be generally considered safe, there is no consistent consideration of all these aspects. And the studies with spike drug substance patches, in our view, are often non-informative on the toxicity, potential toxicity of the impurities contained in these patches. And also the qualification, as is current practice, relies on the traditional use of animal studies without considering non-animal approaches. Next slide, please. So what's the problem statement? The presence in impurity in drug substance and drug products is inevitable. Q3A and B are the main guidelines providing the requirements for qualification. Qualification means providing safety information on the impurity at the proposed specification level. And impurities present in the drug substance, drug product batches at relevant levels in non-clinical safety studies are considered qualified. That's clearly stated in Q3A and B. However, separate qualification would be needed when novel impurities appear or higher specification levels are proposed. Current practice means testing of drug substance batches in animal studies, and such studies never reveal safety concerns and are of little value to inform on the safety of the novel impurity. Guidance on compound-specific qualification of impurity does exist, however, only when it concerns mutagenic carcinogens, solvents, elemental impurities, and in the future, extractable non-leachables. In our view, we need to move away from animal testing of novel impurities and make a compound-specific assessment. Next slide, please. So what's the scope of the reflection paper? It covers chemically synthesized pharmaceuticals when these are not covered by already existing guidances, meaning solvents, elemental impurities, ENL in the future, oligonucleotides, peptides, radiopharmaceuticals. The reflection paper is aimed at qualifying novel impurities or higher level of impurities, which may occur after a change of the manufacturing process or when newly discovered impurities are at place. Non-mutagenic impurities considered qualified when adequately tested in safety studies and or clinical trials. So when the drug substance, drug product has been tested in all these, in a complete program, then implicitly the impurities would be qualified in that case as well. So we are not asking to do this separate qualification on each impurity for any impurity present also in new drug products. And not in scope are ATMP, biological and biotechnological derived pharmaceuticals, herbal medicinal products. And clinical trials is in principle not in scope, since it is not in scope for by Q3A and B, but the principles may be used to guide determination of the level of concern and to provide qualification data if it is considered necessary. Next slide, please. So I give the floor now to Dominic, who will take over from here.
Yes, thank you, Leon. I will give you an overview of the general key consideration for the general outline of the risk assessment. So this general consideration are around four key focus on the 4 key area. The first one is the methodology preference in this reflection paper. So as Leon say, you need to avoid as is possible the in vivo studies, because to be compliant, of course, with ethical consideration and the 3R, and also it would be preferable to use in silico and in vitro method in the new route of assessment. And if this in silico and in vitro method is not sufficient to address the toxicity profile of the impurity, of course, you need to use in vivo studies at least at the final achievement of your profile. That is for the methodology preference. The second key area is that there has been addressed in this reflection paper with some reflection on the exposure assessment. It will be very important to address the toxicity of your impurity in regards of the exposure. First one, we have 2 key considerations. The TTC, you can consider the exposure in regard to the TTC class consideration. So it will be discussed further in this presentation, but also to make this assessment regarding the clinical exposure. And so you need to address, we think that it's better to address that regarding the clinical risk evaluation. The next key area is how to fill the data gap. The data gap. So for that, we address in this reflection paper some line of guideline on the computational toxicology, but also read-across toxicology in order to improve the data and to improve the use of this data in the risk assessment. And so we are regarding the other Q guideline. This is a reflection paper, of course. We are introducing the risk assessment strategy based on the weight of evidence approach that they already use. This kind of strategy as already used in other ICH guideline. But here we try to give you some input of that in order to give more transparency of the justification for the decision of your limit. And so of course what it is very important at this step is this risk assessment of weight of evidence give you a kind of picture. And this is a step for you to decide if we go further or not further for the assessment of your impurity. Next slide, please. So all these key consideration and key area are used in the stepwise process. You are in this reflection paper a flow chart that they made that they describe the way of thinking and to address this the assessment. The first step is to check the applicability of the reflection paper. Of course, you need to determine if the reflection paper apply to this impurity. This is the case if you don't find in the ICH-Q3E and other guideline the way to address your impurity. The second step, if it's applied, it's to assess if the impurity can be qualified based on the metabolic studies. We will have more information further in the presentation on how to do that. And of course, another step very important is to assess and to evaluate, to consider the toxicological concern of the API-derived molecule to see if your impurity it's API-like, non-API-like, and if there is some modification that they could increase the toxicity profile regarding the API of your product. Based on that, you will do an assessment of your toxicological of your impurity toxicological profile, and if after this assessment, the result of this assessment, if you need to fill the gap, you can, of course, fill the data. You can, of course, after look for existing data, relevant data for the impurity toxicity in order to provide your risk assessment, or to use or to find, to identify adequate surrogate molecule, to find suitable surrogate molecule with relevant data. And for that, the reflection paper, isn't a guideline, sorry, give you some input on the use of computational toxicology and approach. Next slide, please. So all this consideration has been drawn in this flow chart. As you can see, you have the first three steps. The qualification, it's applied for qualification for strategy for novel non-mutagenic impurity. You need to see if you can apply other guidelines. After that, you need to check if you have an impurity. It seems to be a metabolite or an API-like, and after you will decide. You have a step to consider the level of concern. This is a risk assessment based on the weight of evidence approach to identify the level of concern and to know if you need to go further or not, and if you need to go moreover. Next slide, please. So just regarding the applicability of guideline, as you know, the Q3E and Q3B is the main guideline, but also I can say the mother guideline for the other Q3 ICH Q3 guideline. So what are the differences? But for the Q3E, you address a mixture of impurity in the drug substance or drug product. For the other guideline, Q3C, Q3D, Q3E, and M7, it's more compound-specific assessment. Regarding the impurity level in non-clinical study, it's qualified in Q3E and B by the NOAEL. In the other guideline, we use also the point of departure for that. In the ICH Q3E and B, there is no assessment. The assessment, we not use assessment factors, safety factors. It's just based on the NOAEL. Sometimes you could have some risk assessment that they use human equivalent dose, but for the other guideline, we calculate a PDE or acceptable intake, and for that we use F1 to F7 assessment factor in order to take in consideration the differences between inter-individual species, but between interspecies consideration, but also on the quality of the data in the animal study. And regarding Q3 and Q3B, you have generic qualification threshold, 0.50 or 1 milligram. For the other guideline, you have not really a qualification threshold. For Q3C and D, it's more based on PDE. Sometimes you have a class of PDE, but except for Q3E under development, you will have a qualification qualification threshold at microgram per day, and for the M7, you know that you have the TTC, but for product where carcinogenic product where the threshold, where we have a relative threshold, you could have a PDE for that. So you have many differences. Next slide, please. Okay. Now we will. I will discuss about the consideration. Consider the level of concern. So next slide, please. So in this guideline, you have a framework in order to address your weight of evidence approach. In this framework, you have 7 criteria. That you can divide in 2 categories. The first criteria is more criteria dedicated to the exposure and the availability of your impurity in the body, and the second one is more criteria clinical criteria indication of the product. Regarding the exposure, of course, some of them are intercorrelate. You have some correlation between of that, but the idea of this the idea of this framework is to give you a picture of the safety profile of your impurity, but also to identify if there is a gap in some information in different criteria, and to give you an input on the need to address this gap and to fill this gap of that. So the exposure criteria, you have the clinical safety margin of the drug substance, not of the product, with different cutoff of threshold up to 110 and 1. And so, of course, if your impurity is there is no safety margin regarding the clinical safety, you are a high level of concern. If not, you have a low level. You have also the daily dose. The daily dose, as Leon said, you have a lot of discussion of the 1 milligrams and the TTC class. So Leon will come back on the TTC, how to address the TTC. You need to consider this point. You have also the route of administration. Is not the same risk if it's an oral, dermal, or if it's a parenteral or respiratory, or moreover intrathecal or epidural route of administration for an impurity. So you need to take that in consideration. And of course, that it's correlated, the bioavailability, of course, is correlated with the route of administration, but you need also to consider the bioavailability of your product, the pharmacokinetics. So if the product have a low bioavailability or high bioavailability, and so you need to consider that in your profile, in safety profile, in the weight of evidence. After, you have the consideration, the clinical consideration to take into account. For example, you have the duration of, for example, you have the criteria for the duration of the treatment. Is depend if you will expose for a long term, or once in the lifetime. Also, you need to take in consideration the clinical indication. The risk the risk benefit will not be the same for S9 product, for example, or life-threatening disease than the convenient regarding the convenient product or OTC product, and of course the existence of alternative product if available. And after, the last criteria is the patient population and underlying condition. It's a rate for adult, for neonate, for children, and for patient with limited liver or kidney function. That we will take that in consideration in your level of concern, and to know if or not you need to be more further for investigation in order to determine your safety profile. Next slide, please. So all these criteria has been discussed in the reflection paper, and so this is a summary of what I say on the assessment of the impurity. So you need to evaluating the risk based on the daily dosage and toxicity threshold. You need to see and to address how assessing the safety margin in relation with clinical exposure level, the target population, to identify if the product is for a specific group like children or pregnant women, the route of administration, and also the duration of the treatment. So next slide, please. I think for the TTC consideration, I will leave the floor to Leon, I think.
Yeah, thank you, Dominique. I will continue here on the TTC. You can find this table in the reflection paper. We have derived and stated TTC levels for the different routes of administration, since the route of administration affects this TTC level. For the oral routes, we currently follow the Kramer class 1, 2, and 3 TTCs, which have been derived in the Monroe paper. These are very old already. However, they are quite broadly accepted also by our European sister organization, the EFSA. That does not mean that this cannot change. There are still efforts ongoing to refine these TTC levels, and perhaps in the future different values or different categories may be defined. For the orally inhaled and nasal drug products, we have proposed a TTC level of 4, which is also quite similar to the OINDP qualification threshold, which has been in use already for extractables and leachables quite a while. That was proposed by the PQRI, which was 5 micrograms per day. We propose to stick to 4 since in the meantime there have also been publications from the Fraunhofer Institute studying a number of compounds and trying to derive TTC levels. And for the more toxic classes, it showed that the range in the range of 2 to 4 microgram per day TTC values could be derived. So we propose here a 4. For the dermal route, the TTC value actually is the parenteral TTC divided by the absorption, since it is the fraction that is actually absorbed that is could be of concern. However, locally, sensitization could be an issue. We consider sensitization reactions as the most sensitive endpoint for local reactions when dermal exposure is concerned. And for this in the literature also, dermal sensitization thresholds have been derived, which are not microgram per days, but microgram per square centimeters, as depicted in this table. For the parenteral route of administration, where a drug is administered directly, systemically, and there is no other barrier that could lead to a reduced absorption or detoxification, it's obvious that there is a lower value for the TTC, and this has been derived by I forgot his name now. Anyway, the references in the paper where 5 microgram is, I think, a conservative approach, but currently the best approach. We are also aware of additional efforts ongoing in the expert working group in the ICHQ3E, and maybe in time when that effort has been concluded, we could also look at those values again. But currently, we propose to use 5 micrograms for the parenteral route. Next slide, please. So if there is a concern and a risk assessment needs to be done, then the first way to do is to look if there are actual data on that impurity toxicological data and use those data for your risk assessment. And to do that, next slide. We propose a methodology which is similar to the well-known PDE calculation methodology, using assessment factors and apply them to a point of departure which is taken from an animal study, or usually taken from an animal study. There are 2 additional assessment factors compared to the PDE methodology that has been described in Q3C. Which is a 6th and a 7th assessment factor, which need to be included to account for bioavailability correction if you have to do a route-to-route extrapolation, or the 7th one if the assessment is not done on the basis of data on the actual impurity, but on the basis of a surrogate compound, which is similar enough to use those data, but not the same. But that introduces some uncertainty. When it is a good similarity, then the assessment factor can be 1. And when it is not so ideal, this factor can go up to 5. And well, when the similarity is not really good at all, then obviously surrogate cannot be used to use that methodology. And a difference, and it was one of the questions we received, what's now the difference between PDE and the acceptable level calculation? Well, there are 2 main differences. One is, as I just explained, these additional factors, which are not described in the ICH documents thus far. And another point is that we consider the calculation of an acceptable level as a level that is acceptable for a specific impurity in a specific product. So it is product specific, whereas the PDE methodology is only compound specific. It is relevant for that specific compound and may be applied to all products across the board, no matter what duration of exposure is at hand for that product, etc. So that's a main difference between acceptable level calculation and PDE calculation. We also propose to use the benchmark dosing limit that will be discussed later on by Roland. And we provided some further reflections on applying the assessment factors in an appendix. However, for the sake of time, we will go not into detail in this webinar on those aspects. Next slide, please. So now I will give the floor to Marianne, who will continue on the metabolites. Thank you.
Yes, thank you, Leon. I will take us back a bit to the beginning of the flowchart to talk about the initial key steps of the framework for qualifying non-mutagenic impurities. And then later I will continue with how computational toxicology tools can be used. But the first step is to consider if the impurity under evaluation is also a metabolite and if it's present in sufficient amounts to qualify the impurity. It's also important to note that the reflection paper only considers acceptability from a safety perspective. So I think if you give it another click, Siska. Yeah, there. So it considers the safety perspective, but not the quality perspective. Next slide, please. Oh, yeah. Thanks. So according to Q3A and B, impurities that are also significant metabolites present in animal and or human studies are generally considered qualified. But as we have defined in the reflection paper, significance should not be understood in the terms of ICH M3, where significant metabolite is observed above 10%, but rather it should be based on the ratio between the concentration of metabolite and impurity. So if the impurity is considered of low toxicological concern, for example, based on literature on the metabolite, it is sufficient that the concentration of metabolite is equal to or above the concentration of impurity. But on the other hand, if data suggest that the impurity is of higher toxicological concern, the contents of impurity can only be considered qualified if a sufficient margin of exposure can be demonstrated. So in the reflection paper, we define the metabolite concentration as the average observed plasma Cmax, and in terms of the concentration of impurity, the daily dose of the impurity calculated as the percentage in the drug product specification relative to the maximum daily dose does not take ADME properties into account for informing on the actual systemic exposure levels, and therefore the daily intake cannot be directly compared to the metabolite level. So instead, a pragmatic approach has been proposed in a paper by Weidolf et al. If you get it a click more, Siska. Yeah, there, thanks. And there, the maximum theoretical concentration of the impurity is proposed to be calculated. So as a worst case approach, the extracellular fluid could be used as the minimal volume of distribution, and different values are given in this paper, which is also included in the reflection paper, for how to then derive the exposure for the impurity to calculate this ratio. Next slide, please. So if the impurity is not a metabolite, the next step is to consider whether the impurity is API-like, or if it cannot be considered to be derived from the API. Next slide, please. So we have included a new concept in the reflection paper to describe impurities that are considered API-like. So that is, they are structurally similar to the API with no new toxicophores while retaining functional groups. This also implies that only insignificant changes to overall structure, size, physicochemical, and pharmacokinetic characteristics introduced compared to the API, which would then be accepted. So the API likeness can be evaluated using computational toxicology and read-across tools such as QSAR models for specific endpoints to assess similarity concerning the structure. So that is if new toxicophores have been introduced, as well as similarities in predicted physicochemical and pharmacokinetic properties. It can also be argued that the dimers or trimers of the parent API can be considered API-like if the dimerization bridge does not introduce a new toxicophore, and if it is systemically degrades to the parent compound. And in the next slide, I have included an example of this. If there's sufficient evidence to classify an impurity as API-like, then no further testing on the impurity is warranted, and the impurity can be qualified using the tox data for the parent compound. But there are some exceptions where minor changes in the structure can lead to an increase in toxicity. So examples would be thalidomide or the lactone form of statins. So this is something that you should be aware of when assessing this, if literature is available to indicate that there could be some concerns, even though the changes would be minor. Next slide, please. So here I've included some examples of when a dimer could be considered either API-like or non-API-like. So in the first example, this is a API sugar dimer where based on QSAR tools, it is predicted that no new toxicophore is introduced, and also that the dimer will degrade to the parent API systemically. So as no new toxicophore is included, it is considered API-like, and then no further data is warranted to qualify this impurity. It would be considered covered by the toxicity data for the parent compound. But in the second example for bisvaliciclovir, this is not considered API-like as the dimerization bridge is predicted to be a formaldehyde releaser using a QSAR tool. So instead, you would need to use formaldehyde as a surrogate for assessing toxicity, and there you could use the PDE value, which is included in ICHM7 and Addendum 2 of 10 milligrams per day, and this value would be taken forward for risk assessment. So this dimer would not be covered by the toxicity data for the API. Next slide, please. So if further evaluation is needed and there's no compound-specific data, another possibility is also to perform read-across to a similar compound with robust data. Next slide, please. So in the reflection paper, we have proposed two different approaches for performing read-across. So either by the single surrogate approach, which includes identifying a robust surrogate for deriving an acceptable level, or for de-risking the target compound, or alternatively using the grouping approach, which includes identifying several similar compounds, which contains the same toxicophores and functional groups that can be used for deriving a group acceptable level or for de-risking the group of compounds. So when performing read-across, a similarity assessment should be carried out that involves chemical and physicochemical and pharmacokinetic properties, where the intention would be to combine the chemical assessment with predictions of physicochemical and pharmacokinetic properties to increase the reliability in the choice of surrogate. Next slide, please. So in this slide, I have outlined the principles of the read-across method. So first, the chemical structural and physicochemical and pharmacokinetic properties of the target compound should be established using either databases or predictive tools. And then the same analysis should be performed for the surrogate compounds to assess similarities for these properties, with the specific focus on identifying toxicophores. Toxicophores can be identified using QSAR or SAR tools describing relevant endpoints, including chronic toxicity for major organs, as well as non-mutagenic carcinogenicity and reproductive toxicity. The choice of adequate surrogates should be justified based on similarities between the different properties and also the uncertainties in the QSAR methods, and the reliability of the outcome should be provided. So if an adequate surrogate can be identified, which allows for deriving an acceptable level, the principles, as already mentioned by Leon, can be used while paying special attention to assessment factor 7, which specifically addresses the uncertainties in the reliability of the chosen surrogate. Next slide, please. But if no adequate substance-specific data is available and if no suitable surrogates can be identified, then it's possible to use computational toxicology tools to build a weight of evidence approach for assessing the toxicological hazard of a compound or to de-risk it. Next slide, please. So several different computational toxicology tools are available for predicting toxicity without the need for experimental animal data. Aside from the read-across approach, there's also several QSAR models available for predicting structure-activity relationships, as well as adverse outcome pathways. Common for these approaches are that they are typically based on broad databases populated with in vitro, ex vivo, or in vivo data, which have been analyzed potentially using machine learning and AI, or where machine learning and AI have been used to train the models to allow for high throughput screenings. So the use of computational tools should always be scientifically justified in terms of the robustness and validity of the chosen model. And it is strongly recommended to use complementary methods across endpoints and methodologies to enhance confidence in the predictions. Next slide, please. There are several commercial and free predictive tools available already to support the assessment, as well as databases that include toxicological and pharmacological data. And as already mentioned, tools integrating multiple data sources enhances the prediction accuracy, and those tools and databases that receives continuous updates also improves the reliability of assessments. There are also several associations that are developing computational tools and have been so for many years. And I've included some of these as examples of the ongoing developments within computational toxicology. So, for example, there's the ASPES cluster, which includes Risk Hunter, Untoxed, and Precision Tox. And this is a European Commission-funded association that continuously works on developing new models for hazard and risk assessment. And there's also the AOP knowledge base that provides an OECD framework for organizing data at the chemical and biological levels. And then there's also the European Partnership for Alternative Approaches that works on developing, for example, quantitative in vitro in vivo extrapolations, as well as physiologically based kinetic modeling. Next slide, please. So in the next two slides, I've included some examples of the use of computational tools. In addition to QSAR predictions of specific endpoints, further data could also be built into the weight of evidence assessment of the compound by using the AOP framework. As already mentioned, several AOP libraries are already available, and here I have included a link to the AOP knowledge base and the AOP wiki. The AOP framework, in short, is used for organizing and presenting knowledge regarding the linkage between perturbation of a specific biological target, which is also called the molecular initiating event or key events, and a consequent adverse outcome. There are a lot of individual AOPs for specific endpoints, but some are also assembled into AOP networks to capture a greater complexity of biological response within the major target endpoints. AOPs have been developed and continue to be developed within major target organs, such as these that have been listed here. And for the purpose of this reflection paper, AOPs are envisioned as a tool to address uncertainties in the QSAR predictions or to fill in data gaps for specific endpoints. So relevant literature from databases could be identified to give information on a specific molecular initiating event or key events to conclude if an adverse outcome is relevant. Next slide, please. So the next slide addresses the use of machine learning and artificial intelligence in computational toxicology. Machine learning and AI provides a powerful tool to develop and validate new computational tools in analyzing data and training models. And I've provided some examples of how machine learning and AI are already being used. So the first example is from Precision Tox, where they're using machine learning and AI to analyze in vitro data and multi-omics to identify molecular toxicity pathways and molecular initiating events leading to adverse outcomes to form new AOPs. And in the second example, this is on the development of a multi-tool that can be used to generate QSAR models, as well as allowing for visual validation of the model. This is based on a 2D rendering of chemical structures using KNIME, and the model is trained to group structurally similar compounds together, making assessment of domain of applicability as well as validation parameters more easy. Next slide, please. So when it comes to computational tools, it's important that the reliability and validity of the used models are sufficiently documented and justified in support of the toxicological evaluation. Several frameworks have been published by OECD on the assessment and review of QSAR models and AOPs, and overall the models should describe a defined endpoint within a defined domain of applicability using an unambiguous algorithm. Furthermore, a thorough review of goodness of fit and robustness and predictivity should be provided. In the reflection paper, several performance metrics have been listed that should be fulfilled to justify that the selected tool is fit for purpose. This could also be provided in the form of peer-reviewed validation studies. I also want to take the opportunity here to highlight the available options to achieve a harmonized regulatory acceptance of a model or a tool via the Innovation Task Force and Scientific Advice Procedure, which also includes computational toxicology tools. Next slide, please. Yeah, so I just included a link to the email webpage for the Innovation Task Force where more information is given. But in terms of assessing, interpreting the results, expert judgment is needed to assess the reliability of the predictions. The usefulness of out-of-applicability domain predictions needs to be carefully assessed, and additional data or results from complementary methods are needed for consensus. The report provided to regulators should provide documentation of the assumptions behind the model, as well as data gaps and model limitations. And by model limitations, I refer to predictions performed with older versions of the tool that needs to be justified that the tool has not undergone significant changes that could affect the prediction performance. Otherwise, it's always expected that predictions are performed using the newest version of the tools. If predictions using computational tools shows a toxicity alert, this should be further investigated using complementary methods, for example, in vitro studies, which my colleague Roland will come back to after me. And as already mentioned, using computational toxicology tools to qualify impurities should be built as a weight of evidence approach, where it's encouraged to combine multiple in silico tools to enhance confidence in predictions, as well as potentially combining with in vitro studies that may be identified as relevant elements as part of an AOP. But I will hand over to Roland to continue the presentation and hope that everything is working with his audio. Are you with us, Roland?
Yeah, can you hear me?
Yeah, perfect. Thanks.
Yeah, and then we solved all the problems. Okay, thank you. So welcome, and I just take over from Marianne, who very nicely and sufficiently lined out the line of strategy towards the qualification of non-mutagenic impurities. And with going over from computational toxicology, I'm further explain in a broad way how the framework is going further with respect of acquisition of toxicological data when it's not possible to reduce the impurity level and the computational and the risk assessment and the level of concern considerations have left uncertainties which need to be fulfilled with additional data. Next slide, please. And with respect to a new approach methodologies, so basically in vitro methodologies addressing specifics with specific toxicological endpoints. There are a lot of in vitro models already out there and described either 2D models there in the Tox 21st project of the FDA and EPA. There are thousands of chemicals have been tested there with 14 or 15 different cellular models addressing a vast variety of different endpoints, measuring a lot of cell toxicity parameters, a lot of signaling, relevant signaling molecules, receptor activation, and other endpoints which are relevant or have found relevant for one or the other toxicological endpoint. And these in vitro data usually, as they are normally used for hazard identification, if you want to go or if you need to go to a quantitative risk assessment, then it's needed then to use an extrapolation via a quantitative in vitro to in vivo extrapolation model. There are also models out there which have been described for the purpose here. And so to extrapolate from in vitro data to in vivo toxicologically relevant concentrations. In the route of the qualification of non-mutagenic impurities, it was already outlined that the reactivity of compound, Marianne has intensively gone over this, is important as a first step here, looking on, and I have here the examples of thiophene, thiourea, protonated nitrogens. Protonated nitrogens have a high probability to bind to the hERG channel, may provide cardiotoxicity. Thiourea are usually carcinogenic within a significant level of concern, and also thyroid toxic. Thiophenes have been reported to be hepatotoxic and also immunotoxic. So this is basically in first approach to look with computational models whether we have a concern here for specific toxicities, and if the computational analysis shows or justifies that additional data to potency data need to be provided for making a quantitative approach, then there and weight of evidence with integrating these data is needed, and there the results from the dose response in those models is then needed to make a potency evaluation. So basically, if there is no relevant response or no positive response in such models, then usually this can be done by a qualitative de-risking approach. If there are findings and relevant results there, then basically in quantitative in vitro to in vivo extrapolation model should be used. And for all these models to be used, it's required that we have some estimation on the validation and validity of these models. Next slide, please. And for the qualification of new approach methodologies, there are various publications from the EMA. There are also some from the FDA and other international regulatory agencies. But basically here I'm focusing on the EMA, and I have listed here the qualification of novel methodologies for drug development, the guidance to applicants, which is already out there for more than 10 years. Then there are essential considerations for successful qualification of novel methodologies. So the guidelines have been also updated during the recent years to reflect the advancement in the technologies, and these guidance documents can be used for providing the justification and validation data for a specific model which has been used to address specific toxicity. For the quantitative extrapolation from in vitro to in vivo, this is needed for the quantitative risk assessment estimation for deriving from this in vitro data, and point of departure which then can be used to calculate the acceptable level with the already shown equation similar to the PDE approach. And there are multiple international projects ongoing. So one is has already been mentioned by Marianne. I've referred to the TOX21 project in the US, where already thousands of chemicals have been tested, and the data are publicly available there from the TOX21 website. And there is this is basically a very good data backup for looking into data and into the models which has been already used there and tested there for the specific endpoints. Next slide, please. And only as a last exit within this reflection paper, where we very much want to emphasize that impurities should be as far as possible and as much as possible qualified by computational read-across and by in vitro models, only if it's not possible to find relevant data in silico qualification models, and when in vitro qualification models provide critical findings which need or which justify the need for in vivo follow-up testing, and also read across is not possible for a specific molecule. So basically, if you have really a new impurity, not API derived, and with no qualification possible by in silico models, then it may be a pass out to then provide an in vivo test or conduct an in vivo test, but there we are emphasizing that it's not scientifically valuable to do in vivo assays only with impurities spiked to a low percentage into the API. You probably will never find the toxicity of the impurity. So it's in this reflection paper emphasized that if in vivo studies are done, then they should be done with the neat impurity and only with those impurities or an exceptional impurity which is not possible to be qualified by in silico and in vitro tools. This also means that the neat impurity needs to be sufficiently available for an in vivo study. Next slide, please. And the in vivo study should be done with the neat impurity. It should be GLP compliant. Basically, it should adhere to the OECD 407 test. But we have some additional deviations for this purpose, and Leon already mentioned that the BMD approach for those response curve calculations and for point of departure calculations is recommended in this reflection paper, and therefore this 28 or 14-day short-term study, if we have only a short-term administration for the compound, is recommended, and basically we consider red unless that is otherwise justified. So red is not the right responding model here. We recommend to use red and both sexes, and the animal dose group and group size should be 3 rats per sex per group, but we should we recommend to use at least 4 dose groups plus a vehicle control. And the reason for that is that usually for BMD calculations, the number of dose groups is more important than the size of the dose group itself. So with 3 dose groups, you may be able to make an BMD modeling, but then the confidence intervals and the precision is not optimal. So with 4 dose groups, you normally get a better precision on the dose response curve modeling and on the BMD and the BMDL. And the doses should be chosen that way that when we just back calculate for the acceptable level, which is anticipated, so depends on what you want to have as specification for the impurity. The highest dose should be calculated with the respective modification or assessment factors that the highest dose provides a suitable exposure margin compared to the anticipated specification level, and the second highest dose should be around that specification level back extrapolated with the assessment factors to then the daily dose in the study, and the lower doses should be at the low no observed level and the low observed level. And the fifth group would be the vehicle control group. Additional, there should be a TK analysis so that we are sure that there is sufficient exposure. The TK analysis can be restricted to the high dose group, and if possible should be integrated into this high dose group to avoid as much as possible to use additional animals for that. Next slide, please. So just briefly summarizing the deviations from the OECD 4.7 here, that's the number of the dose groups, the size of the dose groups, and the integration of the TK analysis only in the high dose group. The rationale, as I already pointed out, is that we are recommending to use benchmark dose analysis for deriving the point of departure. The benchmark dose modeling provides more reliable, robust, and accurate data compared to the no observed effect level consideration. And basically, for the no observed effect level, you have the dose which shows no observed effects, and that is very much dependent on the dose, choosing the doses, and not so much on the dose response curve itself. So there, it's really recommended to make an BMD analysis, derive a proper dose response curve, and set a benchmark response for deriving a benchmark dose confidence interval, and use the benchmark dose lower confidence interval border as a POD. For dose selection, you can also consult the EFSA BMD guidance on the dose selection. I've provided the respective URL here for the EFSA. That's a pretty big document, and provides a really good overview on the benchmark dose modeling, the benchmark dose models itself, and how a benchmark dose analysis should be done. And it's recommended then for deriving the reference point for the acceptable level calculation to use the most human relevant endpoint in the tox in vivo study. That basically usually will be the endpoint with the lowest BMDL. And basically, in exceptional cases, when it can be justified, it may be possible to use an other endpoint which can be justified as more human relevant. Next slide, please. The advantage of the BMD versus the no observed effect level, I already pointed that out, that the NOEL is only the highest dose tested without evidence, and this is affected very much by the dose range selection, whereas the benchmark dose is the dose level estimated from a fitted dose response curve. And important it is to just set the benchmark response. That's basically very often used 5 or 10%, and this is then the relevant point where the confidence interval is then calculated, and usually the benchmark dose, a lower confidence interval level, is then used for POD calculations. This is very nicely shown in the graph there, and there you just very nicely see that the benchmark dose lower level is here derived from the dose range of the study. Next slide, please. And then you can use this point of the point of departure. So basically, the benchmark dose, a lower level or lower limit for calculation of the acceptable level in microgram per day. And usually, there you have for uncertainty an assessment factor of 7 because you are one for assessment factor 7 because you are using an in vivo study, and for bioavailability correction, if you are just using, and that should be used, the route of administration should be the route of administration of the drug the impurity is in. And there usually, if there is not a significant difference for the bioavailability between that and humans, you also have one for the assessment factor 6 here. So it's basically then a PD calculation for the acceptable level as also used in Q3C or Q3D. Next slide, please. Then we are just going to a few comments on considerations for ecology products. They are usually, the impurities are qualified in line with S9. So usually the safety profile is similar to the cytotoxic API. For API-derived compounds, the risk-benefit considerations are important here. And usually there is no need to control limits at levels where no toxicity is anticipated. So this is basically that in vivo qualification of impurities, which are generally not considered relevant for S9 compounds, and therefore these are basically not used and obsolete here. Next slide, please. And for products under clinical development, usually there the ICH M3 is the relevant guideline for the evaluations also of the impurities in investigational medical products, and studies with impurities are generally not required before phase 3. The only exception may be that if there is a level of concern for impurities which guides for additional information, which would be that the consideration level, the consideration of the level of concern will just report that there is an impurity of high concern. So basically, if there is a toxicophore in the impurity which is not in the API and which will given a fire an alert for a critical toxicity, and this is not then possible to be de-risked with computational or other data, then there may be a need for additional studies if that impurity cannot be reduced. I would consider this in very, very exceptional case. I have not seen something like that in my career at the moment. So basically, if the impurity is of very high concern, then there would be additional data needed for qualification. And if so, then the principles and the strategies described in this reflection paper may be used for that. We would appreciate that. Next slide, please. I think that was the last one. Yeah.
Now we would like to discuss the question and answers, or at least the questions that have been submitted in advance. Thank you very much for submitting them. And we have chosen a few of them because quite a lot of questions were submitted. We chose a few of them to respond to. All the others will be responded to in writing as well and will be part of the public consultation feedback document. So thank you again for submitting. They will be answered, and if they're not answered in this webinar, they will be answered in writing in second half of this year when we report back. So then I would like to give the floor to Leon.
Yeah, this question is about the applicability of this reflection paper on products which are on the market for a long time and actually are marketed as well-established use products. Also for these products, the principles of the reflection paper can be used when novel impurities need to be qualified. So yes, it's applicable. Thank you. Well then, this is a question, a level of concern. I thought Dominique was doing this one. Is that correct, Dominique?
You're muted,
Dominique, you're muted.
Sorry, yes, we received two questions on the level of concern. The first one is regarding the high complexity of the feature 2. And so, because there's concern on the multi-factor risk to consider, and each order is considered in the context of the other factors. So the objective of this feature is to provide decision support tools to allowing to visualize the extent of available knowledge across all these multi-criteria. And so by mapping the position of each parameter along the continuum of concern, and they try to integrate, to offer the perspective to integrate the perspective of both knowledge gap, as we have discussed, I've presented during the webinar, and associate knowledge gap associating with the risk level of concern. So normally it's a stepwise where you could have an overview of all the data you have in order to do that. Is not, of course, it help you to take the decision, but at the final step, this is regarding this map, this picture, that you will take the decision to go further, not further, to investigate, to determine the most appropriate risk mitigation strategy, and so that. So this is also something that has been used in other ICH guidelines, like ICH S11. Of course, is for the need of a juvenile animal study to develop the product for a pediatric investigation plan, but also we have this kind of approach also in the ICH S1 BR1 in order to determine the risk, the potential risk of carcinogenicity, of the potential of carcinogenicity, and the need to go to an animal study or not. So this is something that this kind of approach, a weight of evidence approach, this is something that they are already integrated in regulatory guidelines. So I think that after this is a decision tools to support, as I say, the decision. The second question is around about the understanding that we have about acute, subchronic, and chronic treatment, and also if the chronic treatment include the intermittent exposure in this case. So what we will say, we have different definition in the regulatory guideline of acute treatment, subchronic, and chronic treatment. And one of the best definition that we have, this is in ICH E1, where we characterize a long-term treatment, so it's chronic use, as either continuous treatment, but also repeated intermittent treatment administration, lasting more than 6 months. So, as we can say, if you have an intermittent treatment more than 6 months, regarding this definition in this guideline, you can consider that this is a chronic treatment. But if this is intermittent treatment on 1 month and that's all, it is not an intermittent treatment. And so what we, in this guideline also, what we have the definition of short-term treatment is involve only a single dose administration over a few days. And subchronic is, of course, in the sense. So if we can make some definition, acute treatment is a short duration, day or week. Subchronic treatment is lasting several weeks or to a few months, and chronic treatment extended to over months of years, 6 months, included repeated intermittent exposure over long-term source. So we could find this definition in the ICH E1 guideline, but also it has been also addressed in the old ICH S1 guideline. S1 guideline, yes.
Yes, then we have several questions regarding the TTC. Some of them are on this slide. One of the issues raised in these questions is why is there a need to consider impurities down to the level of the TTC, whereas 1 milligram per day has been in use in Q3A and B guidances. And also a reference is made to the paper from Hasselgren that was also shown in one of the earlier slides, and whether we considered those data. Well, I would say that in practice, it's not possible to delineate whether adverse effects in patients are attributable to an impurity or not. Therefore, the argument that 1 milligram per day has been shown to be safe in 3 decades of experience, I think is not a well-founded argument. And the TTC concept, on the other hand, is a well-established toxicological scientific concept and is considered a threshold below which there generally should be no concern. And this TTC level, or the TTC levels, are based on animal data, taking into account differences in sensitivity between animals and humans, and individual differences, and in some instances, also duration of exposure. So we think that there is a scientific rationale to look at levels down to the level of TTC. We did consider the paper from Hasselgren, which I think is a valuable paper, giving us valuable data. And these authors have identified some classes with a high proportion of concerning compounds, as mentioned in the presentation. However, there are also miscellaneous potent compounds not representing specific classes. Also, 2.4% of the compounds have a POD of 0.2 milligram per kilogram per day or below, which only leaves a safety margin of 10. On the other hand, the Graham paper shows that in 18% of the compounds they reviewed, the safety margin is 100 or less, and 53% of those have a safety margin of less than 500. So what safety margin should be safe enough when comparing the NOAEL in an animal study and the patient exposure? Then the last question, it is unclear if this guideline expects lower identification and qualification thresholds than Q3A and B, and suggests that there might be divergence between the reflection paper and the already in use ICH guidance. Well, indeed, Q3A and B define identification and qualification thresholds which should be met from a regulatory point of view. The reflection paper provides the European regulatory view on the scientific rationale to consider impurities when above the TTC. Review of a product on the potential presence of potential genotoxic impurities, as required by ICH M7, could also provide information on the potential presence of non-mutagenic impurities, even when below the identification threshold. It would be a matter of due diligence to consider potential safety concerns of these impurities as well. And it should also be noted that in ICH Q3A and B, it is stated that higher or lower thresholds for qualification of impurities can be appropriate for some individual drugs based on scientific rationale and level of concern. And that is actually what we are asking for. What's the level of concern if you have data that a certain impurity is present, even when it is below the identification threshold? If you by some way already know that it is there, then you would need to consider it. Next slide, please. More questions on the TTC. Yeah, well, the first question is actually similar to what I've addressed on the previous slide, so that's more or less a repetition. And the second question cites a sentence, if exposure level is below the relevant TTC, there's no need for further action, as the TTC levels represent threshold levels for which there's no safety concern. For most, but not all chemicals, the level of concern still needs to be considered in the context of all other aspects shown in figure 2, even when the exposure to the NM is below the TTC level. So that seems to be a little bit contradictory, because the threshold of toxicological concern is defined as a threshold below which there is no concern expected. However, there are always exceptions, of course. It's a cutoff value. And in cases where other factors which have been discussed in this level of concern consideration, but other factors point to a concerning situation, as in the example given in the question, a parenteral drug indicated for lifelong, for non-life-threatening disease in pediatrics, a number of factors indicate there could be a higher concern. Then indeed, even when it is below the TTC, the nature of that impurity needs to be considered. However, it would really be very exceptional if the outcome would be that a impurity needs to be controlled at levels even below the TTC. I think if we have such a concerning compound, then perhaps a methodology not introducing that impurity should be considered. But I think that would really be an exceptional situation. Next slide, please. Yeah, then a question on acceptable level versus PDE and the assessment factors. One question was on if you use these 7 adjustment factors, you could go up to a maximum adjustment up to 10 million or more. Well, I would say that in all cases, all modification factors, if all modification factors would be used up to their maximum value, this would be a clear case where an inappropriate acceptable level risk assessment has been performed. So if the uncertainties are that large and you have to add up all these uncertainty factors and come to a maximum compiled uncertainty factor of 10 million, then the method is not appropriate. And the point of departure chosen is not suitable. so that in practice you would never go up to that such a high level. Okay, next slide, please. Okay.
Yeah, and I take over here. So we received some questions for the read-across approach. The first one relating to whether it's necessary to perform read-across for a metabolite to see if it's similar to the API. So when assessing whether the impurity is a metabolite, if it can be demonstrated that it has been identified as an animal or human metabolite of the API, and that it has been observed in adequate amounts, as I just went through in the slides, then there would be no further assessment needed. So you would not need to perform read-across to relate it to the API structure. And then we also received some questions regarding the requirements for structural similarity assessment. So in the reflection paper, we do not define specific criteria for how to perform this similarity assessment, as it is really a case-by-case assessment. So we do give reference to the ECHA read-across assessment framework that is already in place and has been for some years. And in the reflection paper, we also state that justification concerning any differences in structure, physical, chemical, and pharmacokinetic properties between the target and surrogate compound should be submitted so that it can be demonstrated that the differences are insignificant and does not affect the level of toxicity. But also that a surrogate compound can be considered as a worst case in terms of the toxicity based on these parameters compared to the target compound. So really, it is a case-by-case assessment, but just justifying that the target compound is not of a higher toxicity compared to the surrogate compound. And then the last question here in this slide relates to what I assume is assessment factor 7 for the acceptable level calculations. So in the question, it is asked if the read-across includes multiple analogs, if the correction factor or assessment factor would be affected by this in relation to if you only use the single surrogate approach. So, I mean, if it can be justified that you have several different surrogates in the grouping approach, which would give confidence in the prediction, the read-across method, and that it can be shown that they are robust for performing the toxicity evaluation, then the F7 could definitely be affected and then could be set to a lower level. So as Leon has also mentioned, between 1 and 5, but then going for a lower value if it can be justified that the approach is robust. And the next slide, please. So we also received some questions to the assessment of whether an impurity can be considered API-like. Again, referring to which criteria there would be for performing this structural similarity assessment. So the main point for this assessment would really be to demonstrate that no new toxicophores are introduced in the impurity compared to the parent compound by using QSAR tools and read-across tools, and also that the QSAR assessment should be supplemented with an assessment of key physical, chemical, and pharmacokinetic parameters to justify that the changes in properties, if there are any, would be insignificant and would not be expected to lead to an increase in toxicity, for example, based on similar bioavailability and distribution to cells. Next slide, please. So we also received a lot of questions on the NAM approach and how to use computational toxicology in qualifying an impurity. So it is acknowledged that a single AOP or computational tool may not be sufficient to allow for regulatory qualification of an impurity, and that the computational toxicology right now is more qualitative and hazard-based rather than allowing for quantitative risk assessment. But the intent in the reflection paper is really encouraging using computational toxicology to build a strong weight of evidence case with available literature data combined with predictions of relevant parameters without the need for animal data, so that it can be concluded that an impurity will not be more toxic than the parent compound, that it will not be associated with significant toxicological alerts, so that the level included in the specification can be considered safe for patients based on a lack of hazard for the compound. And if in silico methods are not found to be sufficient for de-risking a compound, the AOP framework can also be used to identify key in vitro studies that describes molecular initiating events or key events, which could rule out a toxic concern for the compound if they are negative. So really, it's looking at the totality of evidence that computational toxicology combined with in vitro studies could give for qualifying an impurity. I think that was the last of my slides.
So basically, this in vivo questions can be more or less summarized by the question or by the concern of stakeholders that because currently, if there is an in vivo testing done for qualification of impurities, then all the impurities are spiked in the API, and then they are using one animal test for qualifying lots of impurities there. It's here for the reflection paper not our intent to that in vivo studies are done for qualifying each impurity. In vivo studies should be only an exception and should only be used if a specific impurity has a high level of concern, cannot be qualified with read-across, with computational toxicology models or in vitro studies. And we consider it that it's simply a very, very big exception that there remains an impurity with an unknown toxicity, with an alert unknown toxicity and no toxicity data. And therefore, this concern is we don't consider the concern here as relevant when you are going the path of this reflection paper for qualifying impurities. The intent here is clearly that you are qualifying this with computational read-across and in vitro methodology, and only in very exceptional cases there may be the need for an in vivo study. This should not so, or the intent here of the reflection paper is that we try to encourage and promote that sponsors are and applicants are trying this path to avoid simply doing an in vivo study, which from a scientific perspective, and Leon has already also pointed that out, that an in vivo study within spiked up API is not providing really valuable data to characterize the toxicity of a specific impurity. And therefore, we want to avoid that such studies are done, and studies are only done in very exceptional cases where the alternative methods we are promoting here and following the framework of here, consider the level of concern are basically not needed. So we do not see here the concern that more in vivo studies are or would be done with each impurity, But if you are following this, and we encourage sponsors to follow this reflection paper strategy for qualifying impurities, in vivo studies are not needed and only extreme exceptional cases. Thanks.
Thank you very much for presenting these questions. Also for the audience, thank you very much for actively submitting these questions. Next slide, please. So please, if you have any remaining questions or comments with relation to this reflection paper, please submit them, making use of the webpage and the public consultation. And we will be very open to your suggestions and comments on this reflection paper. And we look forward to a lot of good comments to improve this reflection paper. Thank you again for attending, and I'm wishing you a great morning, afternoon, or evening. Thank you very much.